Good morning. Today I'm going to speak about the impact of a disturbed environment on a freshwater species, which is the first protagonist of this presentation, whose name is Mauremys leprosa, also called the Mediterranean pond turtle. The second protagonists are those that did the work, and the institutions that we belong to, that are situated in France and in South Africa. Many reptiles in Europe are found throughout the Mediterranean, and this species is endemic to this area. Turtles are one of the most endangered group of reptiles, and Mauremys leprosa is classified by the IUCN as vulnerable. There are several populations of this species in France, but only in the Oriental Pyrenees. Today, we are going to focus in the holobiont, which is the host microbiota assemblage, and more particularly in the importance of the microbiota in the response of microorganisms to the environment. We know that the environment influences our diet and our health. We can consume drugs directly or indirectly through the waters we consume. The environment also influences heredity through epigenetics. And research has shown that the response of organisms to the environment depends on age and on the sex of the individual. In the last decade, research on the microbiota and more particularly on the human intestinal microflora has shown that these processes have a direct influence on its composition. To this end, however, research on wild organisms in their natural environment is still incomplete. And research concerning the study of the microflora of wild organisms in natural or disturbed environments is even more scarce. We therefore try to dig into this question using the Mediterranean pond turtle gut microbiome as a study model. Notably, in one of the most important populations of this turtle in France, situated at La Fosse River, which waters are known to be degraded by the presence of herbicides like glyphosate and AMPA, and by the chemical formaldehyde that is used as a fungicide in agriculture. We further focus on the impact of effluents from the wastewater treatment plant which drains on this river. We captured a total of 11 turtles upstream and downstream of the wastewater treatment plant. We determined the sex, we measured the individuals, and also we did 3 replicate smears of the cloaca, which is the end of the gut and is known to be a good proxy of the gut microbiota itself, at least in birds. We performed DNA extraction and sequencing by using Illumina MySeq that we analyzed using frogs. Alpha diversity, or the number of species in a sample, was measured using parametric and non-parametric indices. Statistical analysis were done using RStudio. Then the differences in the diversity among samples, also called the beta diversity, was studied using qualitative and quantitative indices. Statistical analysis were done using PhyloSeq within R, and different methods to obtain the relationships between samples. Statistical modeling was performed using constrained multivariate analysis, using vegan package in R. And variables analyzed were sex, size, weight of the individual, trap number, as well as location upstream or downstream the wastewater treatment plant. Nearly 2 million sequences were obtained and grouped into more than 200,000 operational taxonomic units, with all the 33 microbiome samples. that were used to calculate alpha diversity indices. Then we perform a statistical analysis, and particularly comparative Mann-Whitney statistical tests showed significantly higher diversity in the gut microbiome of female turtles, with both parametric and non-parametric indices of diversity. Contrarily, all indices except CHA1 non-parametric estimator of diversity was significantly higher for the microbiota of turtles situated upstream compared to the downstream section. If we decompose Shannon diversity into evenness and species richness, evenness is the Pillow index and species richness is the CHA1 estimator index. Because Pillow index of evenness was significantly different between upstream and downstream individuals, but CHA1 index was not, we can then state that downstream differences in the bacteriome of freshwater turtles diversity were due to variations in the number of individuals per species, what is called the evenness, rather than to the number of bacterial species itself, what is known of disturbed communities farther from their climax. When differentiating Shannon indexes among males and females downstream and upstream, we got a surprise. Only the microbiome of downstream male individuals was significantly affected, showing a lower diversity. After normalization and eliminating singletons, a total of approximately 4,000 authors were submitted to beta diversity statistical analysis. As there were no outgroups with this analysis, we averaged over replicates and repeated the analysis. Among all the similarity indices, Morishita index explained the greatest variability of the authors' matrix through PCOAA, principal coordinate analysis. Hierarchical clustering using WWTP method was the most parsimonious of the clustering trees obtained for all distances. And anosine confirmed significance of clustered bacterial communities of females and males upstream and downstream the wastewater treatment plant. Only the microbiome of one male sampled in the upstream section grouped indistinctly with bacterial communities from males located upstream or downstream of the wastewater treatment plant. Nevertheless, when doing statistical modeling through direct multivariate analysis, all models tested were significant, indicating that certain variables explain significantly the variance in the O2 matrix. Distance-based model using Morishita index explained until 60% of the variance with two significant axes and two significant variables that were location and sex. Location upstream or downstream the wastewater treatment plant. All other variables were not significant. To take into account the taxonomic composition of the microbiota according to these variables, we perform a double PCOA analysis. Male turtles downstream were the most diverse in their gut microbiome taxonomy, and they showed a significantly larger amount of Deinococcus thermus phylum. If Proteobacteria, Bacteroidetes, Actinobacteria, or Firmicutes are known to be abundant in animal guts microbiota, and particularly in other turtles, Deinococcus thermus is seldom present in the gut microbiome. This phylum was, however, as abundant as Proteobacteria for this species. This phylum has shown remarkable distance to a range of stressors such as ionizing or UV radiation, oxidizing agents, and desiccation. Finally, with respect to females downstream, they were characterized by a difference in the bacteriodetes number of sequences with respect to females upstream. However, this difference was not significant, and instead we saw a significant variability in the firmicutes sequence abundance between females and in females downstream compared to females upstream. In spite of a small sample size, our results are conclusive. The gut microbiota of Mauremys leprosa is different between males and females and is affected by the wastewater treatment plant effluents. First, with respect to alpha diversity, the female's microbiome appeared more diverse. Turtles' microbiome was less even downstream than upstream, and male individuals downstream had the lowest diversity significantly. Regarding beta diversity, significantly different bacterial communities between upstream and downstream of the wastewater treatment plant and males and females was found. Specific taxa were associated with these same conditions, particularly Deinococcus thermus higher abundance in males needs further research. For instance, a comparative analysis with less stressed Mauremys leprosa population would particularly be interesting. I thank you, the organizers, for having chosen my communication for an oral presentation, and I thank you all the institutions that funded the research, as well as Line Graham for DNA extraction and Genotool for technical support. I thank you, Don, for your attention, and I will be pleased to answer your questions.